All right. Well, it's been a few minutes, and I think we've got just about everyone so far. I will keep adding people in as they come in. So I just wanted to, first of all, say good afternoon, everyone, and welcome. Thank you for taking the time to join us today for our first inaugural webinar as GlobePharma. And we are proud to introduce our ProQbD line, and we're going to focus today on blend and content uniformity. My name is Melissa Herko. I am the Vice President of Sales and Marketing for GlobePharma, and I'm joined by Dr. Sani Raju, the President and Founder, along with Mr. Samir Trika, the Director of Strategy and Business Development. We're very pleased to have such a strong group of professionals joining us from formulation, process development, quality, and manufacturing across all areas and industries. Thank you. Today's webinar focuses on a challenge we all know too well: blend uniformity and content uniformity failures. We all have been there. I've dealt with so many of you that have had that same exact issue. Despite advancements in formulation science and manufacturing, these issues continue to create havoc, impacting product quality, downtime, regulatory outcomes, and untimely time to market. What makes this topic especially important is that many of these failures are not due to a single variable, but multiple different combination of factors, including formulation characteristics, process conditions, and blend performance. So we are going to help you with that with our newest introduction. In today's session, we're going to introduce the DynaSeg, our dynamic powder segregation tester, which is designed to better simulate real-world manufacturing conditions at the formulation level. Wouldn't that be great, right? Catch it early. Before we get started, just a few housekeeping items. The session will run approximately 1 hour. We'll have a live Q&A at the end, so please feel free to put your questions and comments in the chat. We look forward to answering them. With that, I'd like to introduce our speaker, Dr. Sani Raju. Dr. Sani Raju is the founder and owner of GlobePharma Inc., a leading manufacturer of innovative pharmaceutical equipment and quality assurance devices. With a wealth of expertise and numerous U.S. patents related to pharmaceutical equipment design, including the Sift and Blend, and soon to be the DynaSeg, Dr. Raju has positioned GlobePharma as a trusted provider of advanced solutions for the pharmaceutical and nutraceutical industries. In addition to his leadership at GlobePharma, Dr. Raju also founded and served as CEO of Natrion Inc., now a Kerry company, a company committed to achieving near pharmaceutical quality in nutraceutical products. Dr. Raju holds a bachelor and master's degrees in pharmacy from India and a PhD in pharmaceutics from the University of Maryland in Baltimore. He is a licensed pharmacist in New York State and has made significant contributions to both the pharmaceutical and natural product sectors. And with that, I leave it to you. Sani, you have the floor. Sani, you need to unmute. You're muted, Sani. Okay, I think we're having some technical difficulties.
Okay.
You got it?
Thank you. Yeah.
No problem.
Yeah, you can start it.
Okay, well, this is Sani, and I will start our presentation. Please feel free to message any comments or any questions that you'd like to answer and have answered in the live Q&A at the end of the presentation, and we look forward to speaking with you. Thank you.
Sound. Sound किधर है?
Melissa, we don't hear any audio in the webinar.
Okay, there's some technical difficulties with the sound, so I will present this 1 second. Let me fix one item. I apologize. Please confirm that you
Good
can hear
afternoon.
this.
First of all, I would like to thank
Yes, we
all
can hear it.
for
Perfect.
attending this webinar.
AirPods.
We will be focusing on introducing GlobePharma's ProQbD platform of products to address industry's problems of blend uniformity and content uniformity. Content uniformity refers to content uniformity of solid dosage forms. And we have three products in this series. One is DynaSeg, which is newly introduced from GlobePharma. It is a dynamic powder segregation tester to test robustness of powder blends. Second product is Sift and Blend for simultaneous sifting and blending to improve blend uniformity and eliminate the pre-screening step. This product integrates a semi-tubular screen inside the blender with a independently rotating paddle inside the screen. Thus, in every turn of the blender, some material is pushed through the screen. Thus, repeated screening and blending of the powder gives a very uniform blend. And because we have a screen, you don't need to do pre-screening to remove lumps or anything like that. The third product is our sampling devices, which many of you are already familiar with. These samplers are for powders, liquids, and semi-solids to facilitate unit dose sampling and reduce sampling error. Today's topic is DynaSeg, which is the dynamic powder segregation tester. Blend uniformity and content uniformity failures are not uncommon in our industry. We have done extensive research over the last 30 years, and we identified what is causing the real problem, and we present a permanent solution for this problem. This is based on our dynamic powder segregation tester, which has a patent pending. What is content uniformity? Content uniformity dosage forms is critical, especially with low dose potent compounds. For example, fentanyl. The overdose deaths caused by fentanyl are most likely because of content uniformity problems in the tablets. DEA has found that fentanyl tablets on the street have anywhere from 0.05 milligram to 5 milligrams of fentanyl. The fatal dose is only 2 milligrams. That is how important content uniformity is with low dose potent compounds. USP has specification for content uniformity. They have what is called acceptance value. In our experiments, we have used coefficient of variation of the content as at 5% as the upper limit for blend uniformity. We chose coefficient of variation for the sake of ease to compare between the formulations. FDA requires blend uniformity to be tested before proceeding to the next step in manufacturing. What are the factors that influence blend uniformity? One is formulation parameters. Second one is processing parameters like type of blender, whether you use intensifier, what is the speed of intensifier, what mixing time you use, and many other factors come into picture. The third is sampling technique and sample handling. We will discuss these further in the next slides. What are the reasons for blend uniformity failures? First and foremost one is poor formulation. Second one is segregation of the powder blend, especially when it goes to manufacturing. Segregation is difficult to identify at R&D stage because you are making very small batches and segregation doesn't have enough time to develop in small batches. It usually shows up on production scale. Third is sampling from a static powder bed. That is the most critical one. Most of sampling is done from powder blends that are sitting in a blender or in drums after discharge, and sampling is done with samplers, and that gives only apparent content uniformity but not really true content uniformity. This we will discuss at length as we go because that's the main problem that is causing blend uniformity problems. And then bulk powder sampling. Instead of taking unit dose samples, if you take bulk powder samples and send to the lab, the laboratory has to re-sample from that bulk powder, and then they have to transfer the unit dose powder sample to a flask and all that. So in handling and resampling, there would be error introduced. That is another reason for blend uniformity failures. What are the industry's concerns because of these blend uniformity, content uniformity problems? FDA citations for blend uniformity and content uniformity failures. There are many examples in 483 citations. And repeat, and if the blend uniformity fails, then you have to take samples again, test again, and if the content uniformity fails of the solid dosage form, then you have to rework the batch. So a lot of time is wasted, and of course, time is money. The worst case scenario is possible product recall. What is causing these failures? It is really segregation of the blend. What is the solution we are proposing here is first you have to optimize the formulation and the blending process. That is step number one. Number two, sampling from a dynamic and stressed powder bed instead of from a static powder bed. And third, unit dose sampling combined with compaction of the powder samples. This step would eliminate any sampling and powder transfer errors during the process. And then quality by design approach, challenging the robustness of the blends at formulation development stage by subjecting them to dynamic powder segregation test under stressed conditions. We will go over that in detail in the next slides. How can DynaSeg help? Okay, it subjects the blend sample to dynamic segregation testing under stressed conditions of gravity flow, mechanical obstruction with baffles, fluidization of the powder, and vibration. All four mechanisms are integrated into this apparatus, so your powder is subjected to those stresses. Then, as the powder flows through the column, powder collects into a split sample die with multiple unit dose cavities. An integral manual tablet compaction machine allows the unit dose powder samples to be compacted into tablets for analysis. That reduces any analytical error because of powder transfer and such steps in the laboratory. And it also facilitates challenging your powder blend robustness by applying different levels of vibration to the powder while the powder is flowing. This is the apparatus, which I will show more in detail in the next slide. See, the apparatus consists of a stainless steel column, and it has a hopper, so to put the sample here. And there are several alternating baffles, stainless steel baffles in this tube, which would provide the mechanical obstruction to the powder flow. And also, when the powder flows from one baffle to the other, there is fluidization of the powder. And the tube itself is connected to a vibration device which is behind the column here, electromechanical vibration device, and it is connected to the tube by means of a clamp, and there is a sensor there to provide controlled level of vibration, which can be programmed here through HMI, what level of vibration you would like to apply to the tube while the powder is flowing. So, and then we integrated a manual tablet compaction machine in this apparatus so that once the powder comes into the split die here, you can compact those powder samples into tablets using this machine, and you can eject the compacts again using the same machine. There is a pressure gauge here that shows how much pressure you're using to compact the tablets. And this is a hydraulic pump, which is used to do manual compaction. Here, showing a zoomed version, see powder flows through the tube and it collects here. And then you scrape the powder like this. So you have now 30 unit dose powder samples. And those samples are individually compacted using a single punch, or we have a multiple tip punch, which can be used to compact all 30 tablets in a single stroke. This is how the compacted tablets look in the die. And then the top part of the die is removed and placed on an ejection cup, and there is an aluminum pan under the ejection cup to collect the tablets. And again, single punch can be used, and multiple tip punch can be used to eject the tablets from the upper part of the split die. Once ejection is done, this is how the tablets are collected into the aluminum pan, which you can then use for analysis. And I will show you a video clip how this operation works. It is a very simple operation. It takes only a few minutes. this is the HMI where you can program the name of the operators, date, things like that, and then you can pre-select vibration levels or you can manually select. Then you put about 30 cc's of powder in the tube, and it goes very fast and it collects on the split die. And then the die is scraped, the excess powder is scraped with a spatula. So you get 30 unit dose samples. Multiple unit dose samples. It can be a different number depending on the die size, tablet size, etc. That is the multiple tip punch I talked about. And now we are doing the compaction. Compaction is completed. That is how the compacts look inside the die. You remove the top part of the die and put it on the ejection curve, which is hollow actually, and you put aluminum pan underneath to collect the tablets. She's ejecting the tablets now. See, this is the these are the compacts now. So you have 30 unit dose compacts, which you submit to the lab for analysis. And this whole process takes less than 10 minutes, including compaction and ejection. So it goes very fast. And once you submit the compact to the lab, you can use UV analysis or HPLC analysis or something like that to do your analysis for finding, for determining the content uniformity. And you, after this clip, I will show you a little bit more, then in the experiments we have done, we evaluated several factors affecting blend uniformity and segregation using DynaSeg. We have use different types of intensifier bars, a type of blending intensifier attachment used, shift and blend, then the standard high-speed intensifier bar and pin intensifier bar. These high-speed bar and pin bar have been commonly used in the industry, and shift and blend is something we introduced a few years ago. So we will compare these three, see how the results come out. And then we also tried speed of the intensifier attachment. That is a critical step. At 500 RPM, sorry, this is 1000 RPM and 1500 RPM. And then type of blending technique used, whether you are doing dry blending or wet blending, which means are you adding the drug in the form of a solution in the blender and then drying the blend and reblending after drying. And then we also studied drug loading. We have two different levels of drug loading, 0.4% and 0.08%. So these are pretty much low dose formulations because that is where blend uniformity and content uniformity usually cause problems when the dose is very low. The last parameter we studied was particle size of the drug. We have two particle sizes, micronized and USP powder. The model formulation used in our studies is here. We use acetaminophen as a model drug, and we used microcrystalline cellulose 102 by itself or in combination with dicalcium phosphate dihydrate. This is to see when you combine two excipients of different bulk densities and particle sizes, what kind of problems you see. Then the standard ingredients, cross-carbonyl sodium as a disintegrant, silicon dioxide as a glidant, and magnesium stearate as a lubricant. Here is the blending methodology we have used. We used a 4-quart stainless steel V blender. We fixed the shell speed at 25 RPM in all the experiments, and intensifier speed we varied from 500 RPM to 1500 RPM, and 5 minutes pre-blending, 10 minutes with intensifier, and 5 minutes with the lubricant without using the intensifier. Wet blending has additional steps of adding drug as solution while blending and drying the blend in the oven. After drying, the blend is again reloaded into the blender and further steps are carried out then. This is the blender we have used, GlobePharma's Maxi Blend Lab Blender. But here we are showing a 16-quart acrylic shell so that you can see the sift and blend intensifier attachment inside. Here, it's more clear picture with the 16-quart acrylic shell. See, here is the sift and blend. It is a semi-tubular screen which is attached to the hub of the blender. So the screen itself is open at the bottom, okay, and it turns along with the shell at the same speed as the shell. And there is a paddle inside the screen that turns independently of the shell at high speeds. You can vary the speed up to 2500 RPM. So that pushes the material through the screen. In every turn of the blender, some material is pushed through the screen. That is, by repeated screening and blending steps, you get a very uniform blend, and you don't need to do pre-screening for delumping at all. For example, with the silicon dioxide, which is a very nasty material to do screening, you don't need to do pre-screening. Everything happens in the blender itself. And there are 5 patents on this product, and it has been introduced a few years ago. And the other 2 standard intensifier attachments are high-speed intensifier bar, which has discs like this, and it has knives type of arrangement. But on production scale blenders, the design is different. It is not as complicated as this one. The problem with this high-speed intensifier bar is the weight of the bar and the complexity of all the parts and everything. Cleaning becomes a very big problem. And because of the weight on production scale blenders, a lot of people do not use the intensifier bar because it is so heavy and so difficult to install and remove and clean. The third one is a pin intensifier bar. It has, as the name says, it has pins on the shaft. We will we will show you that the pin intensifier bar really didn't work well. Although this worked well, this didn't really work well. We will see that in our results section when we get there. The tablets are analyzed by UV spectrophotometry at 243 nanometers for acetaminophen. If you're using another drug, it may be some other method like HPLC or something. Whatever method you have, you can use that. And the results we will discuss in the next few slides. This is the key for different things. SNB dry refers to sift and blend dry blending. HSI dry is high speed intensifier bar dry blending. Pin dry is pin intensifier bar dry blending. SNB liquid is sift and blend wet blending. HSI liquid is similarly high speed intensifier bar wet blending. Then B in the results, content uniformity results, B refers to the baseline content uniformity, which comes from static sample we have taken from static powder bed. Okay, once the blending is done and the blend is discharged into a plastic bag, we use this a split die and take fill it with the powder, scrape it, and then compact. Those become samples from a static powder bed, which we call it baseline content uniformity. And then GFOB is content uniformity of powder that has been subjected to gravity flow over the baffles. So it goes through the DynaSeg instrument, just gravity flow, no vibrations or anything, and we collect the powder and compact the samples. And then GFOB plus V20, that is gravity flow over baffles with added vibration at level 20. That is number one level, you can consider low vibration level. Then this gravity flow over baffles with second level of vibration. This is with third level of vibration, so that you're really challenging your blend by adding higher levels of vibration to the gravity flow. And the error bars are shown that we have done 5 replicates of each point on the graphs, and we calculated statistical significance using ANOVA, and 0.05 p-value is considered significant. The red horizontal dotted line in the graphs represents 5% CV cutoff mark for acceptable content uniformity. Now we will go and look at the graphs. This is the first one. Effect of different types of intensifier attachments on segregation at 1500 RPM intensifier speed. 0.4% drug loading, and it is dry blending. Okay, here you have 3 graphs. Okay, first one is the green one is sift and blend. The orange one is high speed intensifier bar, and the red one is pin intensifier bar. If you look at these two, okay, the sift and blend and high speed intensifier bar, with both of these intensifier attachments, the blend is pretty robust, because even at increased levels of vibration also, it pretty much stayed below the 5% upper limit for content uniformity. So these two blends are pretty good blends, even with increased vibration, they were holding up pretty good. Okay. But if you look at this one with the pin intensifier bar, it fell apart very quickly. Okay. At baseline, please note at baseline, all three blends, okay, were below nearly 2.5% coefficient of variation, which is excellent. So that is from static bed sampling results. But once you subject these powders to segregation through this DynaSeg apparatus, you can see the pin intensifier bar blend fell apart very quickly. So it is very important to note that, at static, if you do static sampling, they all pass. But when you do real segregation testing, that is when the true colors come out. This pin intensifier bar, really doesn't seem to work well. And then we studied effect of different types of intensifier attachment on segregation at 1000 RPM. Previous one was at 1500 RPM. Now we are studying at 1000 RPM of the intensifier speed. Again, 0.4% drug loading, and it is a dry blending process. In this, we did not use the pin intensifier bar because it really failed at 1500 RPM. So we don't expect it to, at 1000 RPM, it would do worse. So we did not include that anymore. These two are with shift and blend and high-speed intensifier bar. Both of them behaved quite similarly, but again, at static sampling, baseline content uniformity is fantastic, below 1.5%. But the minute we subject it to testing, it crosses 5%, which is our upper limit. So these blends will have a problem when they go to production scale. So at 1000 RPM, we are not getting as good content uniformity as at 1500 RPM. Now we will check at 500 RPM what happens. Again, the two intensifier attachments at 500 RPM speed, same drug loading, and again dry blending. You can see this green line is shift and blend. This is high-speed intensifier bar. So at the low speed, the shift and blend is still better than, is better than high-speed intensifier bar, but both are failing upon segregation testing. Although, again, at baseline, both of them are within the 5% coefficient of variation. But when you test, they go up to even 10% coefficient of variation. With high-speed bar, it went up to even nearly 25% coefficient of variation. So 500 RPM intensifier speed is absolutely a problem. Now this is the effect of drug particle size on segregation at 1500 RPM, intensifier speed, 0.4% drug loading and dry blending. Okay, here this is with shift and blend. We will show the graph with the high-speed bar in the next slide. So this is micronized acetaminophen. Okay, so after all segregation testing at 3 levels of vibration, the CV is still below 5% with micronized powder. However, if you use coarse powder, USP powder of acetaminophen, you see that content uniformity falls apart very quickly upon segregation, dynamic segregation testing. Again, I point out that if you do static bed sampling, both of them passed. So you would be misled that you have a good blend if you do only static bed sampling. You really have to do dynamic stressed powder sampling. Same thing with high-speed intensifier bar, similar results, okay. With high speed also, you see that micronized powder stays within the 5%, even on all stressed conditions of segregation testing, while the coarse powder fails measurably. It went up to 14% here. So, but again, static bed sampling will show that both blends are fine. So you would be misled if you use static bed sampling. Here, it is a combination of fillers. We have microcrystalline cellulose 100% by itself, dicalcium phosphate by itself, and then two combinations. 60-40 and 40-60 of MCC and DCP. So if you see single microcrystalline cellulose filler gives much better blend, very stable, more stable blend than either dicalcium phosphate alone or the two combinations. So when you are combining two fillers for any reason, their particle sizes and their bulk densities have to match. Otherwise, you will have bad blend, which would cause problems in production scale. And then here, the effect of lower drug loading, 0.08% on segregation at 1500 RPM intensifier speed using both shift and blend and high speed bar. So when you reduce the drug loading, what happens? You can see this is shift and blend, okay? So up to the second level of vibration, it held pretty constant. That means it's a very stable blend. But when you increased to the highest level of vibration, then it crossed the 5% level. And with the high speed bar, you can see that it was going up and it crossed the 5% at the first level of vibration. So I think marginally the shift and blend is probably doing better than the high speed intensifier bar. Here it is wet blending. Adding the drug in the form of a solution. We add the drug in solution, blend it, take the wet blend out, dry it in the oven, and put it back into the blender and then complete the blending. So with this, you can see that both shift and blend and high speed bar both gave pretty similar, almost identical results. And at any of the vibration levels, it's pretty constant below 2%. So that is what you would expect when you are doing adding the drug in the form of a solution, because you are distributing the drug at molecular level, so you get very uniform distribution, and the blend is very robust. This is the kind of blend that would never ever give you problems on production scale because it is so robust. It is a wonderful formulation. And also, see, this graph shows that the DynaSeg dynamic powder segregation tester is working. It shows that the blend made by wet blending is very robust. It is very clear that the powder segregation tester is really, really working well. And let us talk summary of the results here. Uh, Dynamic segregation testing under stressed conditions of gravity flow, mechanical obstruction to flow, fluidization, and vibration, all combined, reveals static powder bed sampling gives only apparent blend uniformity, not true blend uniformity. You would be misled if you use static powder sampling. Most formulations failed blend uniformity specification under stressed dynamic conditions, which are characteristic of large-scale manufacturing, although they all passed blend uniformity specification when samples were taken from a static powder bed. Dynamic testing also revealed that the formulation variables such as particle size, excipient bulk density, and drug loading, as well as process parameters such as intensifier type, intensifier speed, and blending method strongly influence segregation potential. Addition of controlled vibration during testing further challenges the blend robustness and meets QbD guidelines. A robust blend avoids content unit problems in manufacturing. That is your goal. Once you make a powder blend and put it on a tablet machine or encapsulation machine or powder filling machine, you should be able to go and sleep. So it should be absolutely problem free. What is uniqueness of this apparatus? It combines 4 different mechanisms of segregation: gravity flow, mechanical obstruction to flow, such as, let me go back. Gravity flow represents flow of the powder from a hopper onto the tablet press into the force feeder or gravity feeder. And mechanical obstruction to flow is a representation of the powder going through a force feeder where impeller is moving the powder, so there is a mechanical obstruction there. And then fluidization, when the tablet press is running very high speed, there are centrifugal forces, and also during powder flow from the hopper, there may be some fluidization. And there is vibration. The machine vibration is there, so that also would contribute to segregation. So it combines all 4 mechanisms of segregation into one instrument. And the test requires a small sample size, about 30 cc for each run. In some of the literature, we have seen some of the models people have developed. They require a large sample size. So that is another inconvenience with, when you need, 2 kilo, 3 kilo sample size. And it takes less than 10 minutes to run a sample, including sample compaction and compact ejection. We get very reproducible results, as we have seen in the graphs. We have done 5 replicates, and when the blend is uniform, we get very good, reproducibility. And this apparatus can be enclosed in a glove box if containment is needed. And it's very easy to clean between the runs. After each run, you just vibrate the column, so any loose powder will come out. before you start the next test. And, extensive cleaning, you can take the tube and the baffles out and clean them and dry them and put them back. In conclusion, dynamic powder segregation testing should be made part of formulation and process optimization to avoid costly content uniformity problems in manufacturing. So you should think of doing dynamic powder segregation testing as a part of optimization of your formulation and process at the R&D stage. That way, when your formulation and process are transferred to manufacturing, they will not face any problems. Thank you all for your attention. And the next webinar will be probably in May on the second product, which is shift and blend technology. Please join that webinar as well. And thank you all once again.
Thank you, everyone, for attending our webinar today. I would like to open it up to see if there were any questions or any additional information that you were looking for. I don't see anything. Anyone?
You can submit your questions by email. We can answer that way, and the link for this presentation will be on our website soon, so you can always go back and go through that when you get a chance. I know some of you joined late. So you can go back and watch that, and feel free to send any questions you have. We would be glad to answer. And our goal is to make you successful in what you do. And please feel free to come back to us if you have any questions. Thank you all.
Before we say goodbye, I'd like to introduce our Director of Strategy and Business Development, Mr. Samir Trika. If you'd like to come on camera and say hello.
Thanks a lot, Melissa. Hey, everyone. Thanks a lot for your attendance and attention today for the webinar. I recently rejoined GlobePharma about 2 months ago, as Melissa mentioned, as the Strategy and Business Development Leader. Really looking forward to working with all of you, and thanks again for your attendance. And we look forward to the next presentation in May about the shift and blend technology that Sunny reviewed partially today. Thank you.
Thank you all.
We will be sending out a link with our email to say thank you, and it'll have download for all of the presentation. And if you require any additional information or additional presentation direct, please feel free to give us a call or to email us back, and we would be more than welcome to set that up for you. Have a wonderful day.
One quick final note. You have seen that the pin intensifier bar didn't work, and the low speed of intensifier didn't work. So you can imagine if you are doing blending without any intensifier bar, what kind of results you would expect. If you're doing blending without intensifier and doing static bed sampling, you're going to be misled that you have a good blend. So please pay attention to that, and please shoot us any questions you have. Thank you.
Have a wonderful day.
Bye-bye.
Bye.
Bye.